In my last class, I had discussed the structure and functions of immunoglobulins. Now, today's class is going to deal with complement and activation of the complement by three different pathways. But since one of the pathways involves antigen-antibody interaction, I thought I will deal with a little bit of the structure of immunoglobulin, so that one can understand how complement can be activated through this pathway. Now, I had also forgotten to mention a few important molecules while talking about the structure and functions of immunoglobulin, and I will also in very brief describe those. Now, let us first look at the structure again of immunoglobulin. This is the prototype structure, as you know, and you might remember that this is the prototype structure for IgG, also IgD and IgA. IgE and M have a modification of the region called the hinge region, and this is what I would like to describe. In IgE and IgM, the hinge region is modified into a domain itself, and therefore, only these classes of immunoglobulins have, apart from the three constant domains, one extra constant domain. So, the hinge region now becomes CH2, then there is CH3 and CH4. Anyway, is there anything specific with regard to hinge region? It was found that the hinge region has a large number of the amino acid proline. Why proline? Is because the addition or inclusion of this amino acid lends flexibility to the primary amino acid sequence or the polypeptide chain. Now, why should there be in the hinge region number of prolines which gives flexibility to the molecule? Because we do know that immunoglobulins are at least bivalent. Now, monomeric immunoglobulin molecule, which is made up of 2 heavy and 2 light chain, now can bind to 2 domains or antigenic determinants of an antigen, 2 at one time. Now, if one is occupied, the other one may experience steric hindrance, by which it will not be able to make contact with the another molecule of antigen or the same epitope on the same antigenic structure. Therefore, nature has it that the hinge region is very flexible and allows sufficient movement of the 2 Fab molecules for efficient binding to antigen. So, that an antibody molecule can be at least bivalent in its function. I had already mentioned to you that if you take the heavy chain separate from the light chain, that is, it is an easy experiment, one can use DDT or any reducing agent like beta mercaptrethanol to break the disulfide bonds which hold the 2 heavy chains as well as each of the heavy chain to one light chain. Now, this can be after reduction of these bonds, you can have 2 heavy chains and light chains separate. Experimentalists have tried to see whether the light chains can bind to the antigen when separate from the heavy chain. Now, it was seen that at higher concentrations of the immunoglobulin, reduction leads to precipitation, and this heavy chain, as I probably mentioned to you, heavy chain alone, if soluble, can bind to antigen. The light chain usually does not show any binding to antigen. We do know that binding of the immunoglobulin molecule or the antibody molecule to the antigen is more dependent on the structure of the heavy chain. Now, like I said that heavy chain separated from light chain, though it can bind to the antigen, the affinity of the binding comes down tremendously. It could be 10-fold or even 100-fold. In any case, one needs to think in terms of the solubility of the molecules. Like I said, at higher concentration, the immunoglobulin chains, they precipitate, and of course, heavy chain more likely than the light chain. Now, what is the reason for that? That is because the coming together of the heavy and light chain, to form a structure which can bind to the antigen, this interaction is through hydrophobic surfaces. So, when these two chains are separated, the hydrophobic patches are exposed, and this will bring about aggregation of the self molecule, that is, two heavy chain would come together, and this would bring about precipitation of the molecule. So, therefore, expression of immunoglobulins in recombinant systems, especially prokaryotic system, is rather difficult. Now, why did I say all this? Is because in the sometime in the late 90s, there were reports on a type of antibody which is specifically found amongst mammals, found in camel group of animals like dromedary and camel. Now, interestingly, it was found that 33% of the immunoglobulins in camels is made up of two heavy chains only. But just to recapitulate your memory, that it is two heavy chains and two light chains which comprise the classical antibody, and the N termini of both these chains makes the antigen binding pocket. In case of camelid antibodies, which again I will like to stress is only around 30 to 33% of the total immunoglobulins found in the camel, but these are made up of, they are much smaller, and they are made up of just two heavy chains, which are of course identical. Just to tell you, we do not deal with the lower vertebrates, and whatever immunology we discuss is that described in mouse and human. But I would like to tell you that sharks also have similar molecules. Of course, the repertoire is much restricted in sharks. Now, the camelid antibody is made up of two heavy chains. Now, this would mean that there had to be some kind of modifications in the camelid antibody, so that it could do away with the light chain, because all along we have been learning, that both heavy and light chains come together and they form the antigen binding site, and therefore, how does the heavy chain in camelid antibodies manage without the light chains? Because like I said, the repertoire appears to be the same in the camelid antibodies compared to the classical antibody. Now, I am not going to deal with the entire paper that was described by two scientists who are from Belgium, but I thought I will just tell you that there are what could be the main two modifications that the camelid antibody chains needed to undergo, so that they now become soluble protein and are effective. To compensate for the light chain, the hypervariable domain of the heavy chain is enlarged by 7 to 8 amino acids. Importantly, the hydrophobic patches on the heavy and light chains that are involved in hydrophobic interactions with each other are changed to hydrophilic regions. So, hydrophilic amino acids would not bring about this cascade of precipitation as one sees when the classical antibody is reduced to separate the light and heavy chains. Interestingly, this camelid antibody is very stable, and it maintains function even at 100 degrees centigrade and also extreme pH treatment. Now, camelid antibodies, looking at the structure of camelid antibodies, looking at the sequences, has actually provided immunologists to start thinking in terms of modifying the classical antibody at the gene level, so that one could probably get antibodies as recombinant proteins and just the heavy chains, which would be much simpler than if we were to clone and express both heavy and light chain genes into a prokaryotic system. So, therefore, camelid antibodies have provided enough of interesting ideas to biotechnologists. There is another set of antibodies which I thought I should introduce to you, because in the last 20 years or so, IgY antibodies from the avian system are being utilized in diagnostics. Why IgY antibodies? Now, IgY is an antibody that is found in the avian system, and chicken or hens are now recruited for this kind of, generation of this kind of an antibody. Why is it interesting? Because in hen, it has been seen that circulatory proteins are deposited in the egg yolk. Therefore, in immunoglobulins, these antibodies are also, when a hen is immunized or in a natural condition is exposed to pathogens, the antibodies that are generated are automatically deposited in the egg yolk. The immunoglobulins are taken across the oocyte membrane and deposited. Therefore, now the egg becomes a storehouse of these IgY antibodies. Interestingly, one can get 3 grams of immunoglobulin Y per month per hen. So, that becomes also an extremely cheap way of getting large amounts of antibodies. Immunizing hen is not any more difficult than immunizing rabbits. Also, most importantly, apart from the commercial aspect, no cross reactivity is seen between IgY and mammalian immunoglobulin. Therefore, there is no interaction with mammalian Fc receptor. Therefore, there will not be any interference, especially when IgY antibodies are used in diagnostics. So, apart from camelid antibodies, IgY antibodies also have a very interesting commercial aspect. So, now we come to the complement system. Complement activation is an important or an integral part of the humoral immune response, and it is an integration between the acquired immune response and the innate immune response. The complement system comprises a very large number of soluble and cell bound proteins. There are more than 30 soluble and cell bound proteins in this system, and like I said, these impact both innate and acquired immunity. What does the complement cascade do? Now, amongst all the functions of the immunoglobulins, you might recall that immunoglobulins or antibodies bind to their cognate antigenic epitopes on antigens in a very specific manner. Antigen antibody interaction it activates the complement cascade, and we will be dealing with the mechanism part. But what does this activation of the complement system, whether it is classical, alternate or lectin, what is the ultimate result of the activation? It is the pathways forming membrane attack complex, which now induces lysis of cells, bacteria, viruses, also causes opsonization, inflammation, importantly clearance of immune complexes. So, we will be dealing with all these when we look at the activation of the complement cascade. Complement cascade or activation culminating in the formation of the membrane attack complex can be antigen and antibody interaction dependent, as happens in the classical pathway, or the alternate pathway and the lectin pathway. Now, the alternate pathway and lectin pathway are both systems of the innate immune response. Always good to know a little bit of the history. When were these complement components discovered? They were first, the complement system itself was first described in very brief around 1890s by a scientist, Jules Baudet. Now, what was his observation on which he based the or described the complement? Now, he had made sheep antiserum to the bacteria Vibrio cholerae and found that under certain conditions when the Vibrio cholerae or the bacteria were incubated with this antiserum, there was lysis of cells. Now, he found also that this was when this antiserum collected was rather fresh or rather recent. Therefore, now he chose to heat and activate the serum and found that about at 58 degrees, heating the antiserum at 58 degrees did not abrogate the reactivity or agglutinating activity of the antiserum to the bacteria, but it abrogated the lytic activity. So, therefore, he knew then that there was a component in the serum apart from antibodies which is able to bring about lysis of cells. It was later, couple of years later, that similar observations were made by the scientist Paul Ehrlich, and he called this complement. Complement because it complements the activity of the serum. Now, even before we come to the complement components, let us see where are these complement components synthesized. Mainly by the hepatocytes, also by the tissue macrophages, and by the epithelia of the gastrointestinal tract and the genitourinary tract. Complement components are quite large in number because 5% of serum globulins are constituted by complement components. All the complement components circulate in the serum in a functionally inactive form. You can call them zymogen. Activation of the complement leads to cleavage, and the cleavage can be A smaller fragment and B larger fragment. Of course, there are some differences in the size of the fragment and which one now becomes the active complex of the complement cascade. But the complement components, and I will be dealing with that in detail and in a sequential fashion, complement fragments, they interact with each other to give functional complexes. Now, I will just like to tell you all about So, I will just like to tell you all about all complement components. There is a common strategy. So, you have now, let us say this is the one of the components. After this gets activated, and all these are usually serine proteases if they are enzymes. Now, when a complement component gets activated, and I will tell you how the activation happens, then there is usually a cleavage. There is usually a cleavage. Let us say this one is C4. Now, the larger fragment, which would be C4b now, and the smaller fragment C4a. Now, the larger fragment gets associated with a component which is already present at the site of the activation. So, you have now C4b which would come and bind here. The binding of these two would bring about a complex which may have an enzyme activity. Now, whenever a complex has an enzyme activity, such a complex is denoted by a line above the components to say that this one is an enzyme. Now, I will come back to my So, we will come back now to the activation of the complement cascade. So, the example that I have given here, complement fragments interact to give functional complexes, for example, C4b2a or C3bb. As such, on their own, they are not enzymes. When they come together, they have specificity with respect to a particular substrate. Let us first come to the first component the first pathway, which is activated by antigen antibody interaction. The classical pathway has components which comprise of C1 to C9 different components. Because the classical pathway is activated by an antigen antibody interaction, we will need to see how this actually brings about activation of the molecules. Now, binding of antigen with antibody induces a conformational change in the Fc region of immunoglobulins, and just to recall, the two classes of immunoglobulins that can fix and now activate the complement components are IgG, and only 3 classes of the subclasses of IgG and IgM. So, in these two, there is, upon antigen binding, exposure of the C1 binding sites on the Fc region. Now, the C1, what is C1? The C1 itself, which is the first component of the classical pathway, is itself a complex. It is made up of Q, R and S, and these 3 are held together as a complex which is stabilized by calcium. So, without calcium, there is no activation of the complement cascade. Let us look at first the C1q. This is the molecule C1q. As you can see, this is a flowery structure which is made up of, actually, which does not show here in the diagram, but it is made up of 18 polypeptide chains, which now are held together with one part which is like petal, and 3 of such polypeptide chains come together to give one such head. There are 6 heads. On the other hand, the C1s and C1r are complex together very closely, and in the serum they are usually present as an S-shaped complex. But the same can bind to the C1q in a calcium dependent manner, and when bound to C1q, they have a structure which is H-shaped. In the serum, of course, this complex is not active. Let us look at what happens when the complex is recruited to the site where an antigen antibody interaction is taking place. Now, the C1 component, which is made up of 3 molecules, is recruited to the site where an antigen, which is, let us say, a component of a bacterial cell. Now, this is a bacterial cell, well, half a cell. Okay. Now, you can see that there are specific antibodies which are these black shaped Y-shaped structures which are binding to specific antigens present on the bacterial cell surface. Though I have put the antigen in the same color, it need not mean that all the antibodies here should be identical. There could be 2 different antibodies recognizing 2 different antigens on the bacterial cell surface. Now, the C1q binds to an exposed region present on the Fc region of the immunoglobulin molecule after it is bound to the cognate antigen. So, it is very important to know that antigen antibody interaction brings about a conformational change not only in the antigen binding site, but also the Fc region, which is the constant domain of the immunoglobulin molecule. There should be at least 2 heads of one C1q molecule such that it can bring about activation of the R2, S2, both of which are serine proteases. Now, it is also important here to make you remember that IgM is a better activator of the complement cascade. If you remember the table that I showed in the last lecture, IgM is much more efficient in activating the complement cascade as compared to any one of the subclasses of IgG. The reason being that IgM is a pentameric structure, and since the two heads of one C1q molecule is required to activate the complement cascade. IgM, which has a pentameric structure, is more likely or to be able to involve or engage two heads of the same C1q at one particular position. Therefore, IgM is much more efficient. Now, what happens here? Here of course, I have only IgG, and let us go on with that. So, at least two immunoglobulin molecules should be in close proximity, so that two heads of the C1q molecule get engaged. The binding of the C1q to this exposed site on the Fc region now brings about a conformational change or induces a conformational change in the C1 molecule. The C1 molecule, upon its conformational alteration, induces a conformational alteration in the molecule that is closely associated with it, that is R2. The R2 now activates S2 by chopping off or cleaving a part of the S2. The S2 is an enzyme and has two substrates. One is C4 and the other one is C2. So, remember, all this is happening on the surface of the bacterial cell, and though the various activities that I have shown in pictorially, all this is in fact happening at the site of the antigen antibody interaction and the C1 complex which is binding to it. So, everything happens at this position, but to make it clear and to think in terms of the sequence of events that are taking place, we draw the or depict the antigens far away from that of the antigen antibody and the C1q binding to the antibody molecule. So, C1q binds to the Fc region of the immunoglobulin, bringing about a conformational alteration, which is now passed on to the R2, activating it, which in turn activates the serine protease S2. The cleavage of S2 to its active form also now exposes a binding site for the second molecule in the sequence, which is C4. Now, though the complement components of the classical pathway are made up of C1 to C9, the sequence in which they are recruited may not necessarily be so, and this is the part where after C1 the next complement component to get activated is C4. Now, C4 is recruited to S2, which cleaves now, which remember by now it is activated. So, it cleaves C4 to C4a and C4b. C4b is the larger fragment and gets associated with S2. C4a on the other hand is the smaller component and diffuses away. Now, I will come back to what C4a does, but let us come back to the C4, which is now bound or in close proximity to S2. Now, S2 recruits yet another component, which is C2. C2 is cleaved by S2 again. I told you S2 has two substrates, C4 and C2. Now, C2 gets cleaved to two fragments again, C2b and C2a. C2 is the only molecule now in this particular category of molecules where the C2b or the larger fragment diffuses away, whereas the smaller fragment C2a now complexes with C4b, and this complex, which is stabilized by calcium again, is an enzyme which is C3 convertase. And you can see that the complex is now, because it is functionally active, is denoted by a line which is across the C4b2a name. Now, C3 convertase, as the name suggests, recruits C3, which is yet another protein in the serum, again in its inactive form. C3 binds to C4b2a, the C3 convertase, gets cleaved to a larger fragment C3b and a smaller fragment C3a. the C3A diffuses away and C3B binds to C2. This now binding of C3B to this C3 convertase now changes the specificity from being a C3 convertase to a C5 convertase. So, C4B 2A 3B, well actually it is C4B C2A C3B, but since C is common, it is written as C4B one component, 2A another component and 3B. The C5 convertase, as the name suggests, has a binding site for the next component, which is C5. C5 convertase now cleaves C5. Now, just remember, every time there is some activation, like in this case, there would always be a conformational alteration, like for example, in this case binding of C3 to the earlier components now changes or makes accessible a binding site for the next component C5. So, the C5 is therefore recruited because there is now a binding site on the complex, and this is cleaved from C5 to C5A and C5B. C5A again diffuses off. Now, at this juncture I would like to tell you C4A, C3A, C5A, all these three are anaphylatoxin, and we will be dealing with this in detail in the hypersensitivity context. But these are molecules which induce inflammation and inflammatory response through mast cells, basophils as well as eosinophils. There are specific receptors that bind to these components. So, they have use. C2B on the other hand is a question mark. What does C2B, what does it do is still a question mark. Now, let us get back to the complex. We have now C5B, which is formed by the enzymatic cutting or cleavage of the whole C5 molecule. What happens next? Now, C5B has a tendency to bind to a surface membrane, but it is not very stable, and it is not stable unless, and starts to degrade unless the next component, which is shown in yellow here, C6 binds to it. The moment C6 binds, C5B, which is held loosely on the membrane, gets stabilized. The binding of C6 to C5B brings about a conformational alteration in the molecule C6, so that a binding site for the next component C7 gets exposed. This complex now can recruit the next component, which is again in sequence C8. One C8 molecule is recruited to the already existing C5B, C6, C7, which now, this is the complex here, you can see that there are 4 components, each one in different color. Now, this is an important step because this component now undergoes a change from hydrophilic to a hydrophobic status. Why is this required? Now, what I mean by this is that in the serum, all proteins need to be soluble. When they come now onto a membrane and they need to be anchored on the membrane, they need to have hydrophobic surfaces or regions exposed. So, this entire complex now, when 6, 7, 8 are bound to C5B, undergo a hydrophilic to hydrophobic transition. So, that now this complex can get into or anchor in the lipid biolayer. The moment that happens, the next complement component C9 molecules are recruited to the site. C9 on its own is present in soluble form, but when the binding site for C9 is exposed on the C5b 6, 7 and 8, there is polymerization of the C9 and there are large number of C9 molecules which are recruited, and you can see this forms a structure which is known as membrane attack complex. The polymerization of C9 and the hydrophobic activity of the C5b 6, 7, 8 brings about a pore forming complex, which is called, like I said, membrane attack complex, or MAC in short. Now, what does MAC do? As the name suggests, it lyses cells. How will it do that? It actually makes a pore in the membrane. One MAC is adequate to lyse one red blood cell. However, in case of lysing nucleated cells, more than 3 MACs are required. Gram-negative bacteria are susceptible to MAC-induced lysis, whereas gram-positive bacteria, which have peptidoglycan layer, are resistant. I have already talked about these complement components. Let us just go over them in brief. So, the classical component pathway proteins are made up of first the initial components, which are all enzymes, except of course C1, but the C1q complex is C1, which is made up of C1q, C1r, C1s. C1 starts or initiates by binding to the Fc region of IgM or IgG. Upon getting activated in such a way that is by conformational change, it induces the serine protease C1r to activate C1s, which is also a serine protease. Now, what are serine proteases? Those proteases which have serine in the active site. So, all of these are more or less same in terms of classification of enzymes. Now, C1s, it activates C4 and C2. So, substrate for C1s is C4C2. C4, which is made up of C4a and b. C4a is a peptide mediator of inflammation. C4b binds to C2 and the complex is then cleaved by C1s. Now, C2a is a serine protease and C2b, the function is unknown, and both these are a part of the C2. When C2a binds to C4b, it forms now an active C3 convertase. C3, which is one of the most important molecules of the complement components, again is made up of two C3a and C3b, or rather C3 gets cleaved to these components. Some of them have more than one subunit, but we will not go into that. The C3 gets cleaved to C3a by the C3 convertase, which is a peptide mediator of inflammation. The C3b on the other side, or on the other hand, binds to the C3 convertase to form now the C5 convertase. Proteins involved in the formation of membrane attack complex are C5, C6, C7, C8 and C9. The active forms are C5b and the others only after they bind to the components which are activated before, which is C5b. Now, it is at this stage C5b 6, 7 that there is a transition of the soluble component to become now amphiphilic, which will have hydrophobic surfaces inserting into the lipid bilayer. C9 of course polymerizes to complete the formation of the MAC pore. Now, there are some reports that say that in the absence of C9, in some cases just the complex formed between C5, C6, C7 and C8 is enough in fact to kill a cell. Now, let us come to the alternate pathway of complement activation. Like I told you before, the alternate component is a part of the innate immune system. Now, so is the classical component, except that the classical component or the cascade is initiated only upon antigen-antibody interaction. Let us look at now what is the pathway of the alternate cascade. Let us start off with the first component in this, which is C3. Now, the molecule C3, which is present in the serum in an inactive form, has a labile thioester bond, and therefore, there is auto conversion to C3 and C3b, but this is very slow process. But since it's C3 is in the serum, and since there is this autocatalysis, there is always C3b, C3a formation continuously in the blood, and if there are bacteria in the blood, this cleaved C3b binds to the bacterial cell surface. Now, does C3b bind to even mammalian cells? Yes. In fact, C3b can bind to all surfaces, all cells, but on mammalian cells, since the surface is sialic acid rich, this molecule gets degraded very fast. Bacterial surfaces, on the other hand, have very few sialic acids, almost none, and therefore, C3b binds a little bit more stably. Now, C3b bound to the surface recruits a factor B. C3b bound to the cell surface has a binding site for another serum factor called factor B, and this binding is magnesium ion dependent. If this complex is not now bound to factor D, in fact, this complex is also not very stable. But when factor D is recruited to this site of the complex factor B C3b, factor D, which is a serine protease, cleaves factor B to factor BA and BB, and this is where we come from here. So, you have now C3b, which is bound to cleaved BB. Now, factor D has its substrate is factor B. Once B is cleaved to BA and BB, which is bound now with C3b, this becomes a C3 convertase. Now, this needs stability, and unless there is another serum protein called properdin, which binds to this complex and makes it stable, this is highly unstable complex. After stabilization of properdin, the complex C3b-BB, which is a C3 convertase, now recruits C3. Now, C3 is recruited to this site, and the C3 convertase converts C3 to C3a and C3b, and now C3b is recruited to this site, which becomes a C5 convertase. So, you have C3b-BB, which is C3 convertase, and you have C3b-BB and C3b. So, now C5 convertase has 2 molecules of C3b, 1 molecule of BB. As the name suggests, again C5 convertase recruits C5 because there is a binding site on this complex where C5 binds, and the C5 convertase cleaves the molecule to C5a and C5b. Now, the rest of the sequence of events is identical here on between the classical component and the alternate component, because you have formation of C5b, which recruits C6 first, and the binding of C6 to C5b brings about a binding site or makes accessible a binding site for C7. This undergoes along with C8 a transition to a hydrophilic or amphiphilic structure, which can insert itself into the lipid bilayer, thereon recruiting C9, because there is exposure for on this complex for this polymer of C9, and you have the formation of a pore forming complex called MAC. What are the alternate complement pathway proteins? They are C3A, C3 mainly, because the complement cascade actually starts with the auto conversion of C3 to C3A and C3B. Now, remember that C3, there is a fast way of getting C3 converted to C3A and B, and that would be by the enzyme C3 convertase. But in the serum, a slow reaction is always taking place, where C3A and C3B are formed. C3B binds factor B, which is now cleaved by factor D to give C3B BB. The C3A, as in discussed earlier, is a peptide mediator of inflammation. Factor B, which is also a serum protein, gets cleaved by the factor D to BA and BB. The BB complexes with C3B to make the C3 convertase, and then recruits another C3 molecule, so that there is generation of a C5 convertase. Factor D binds to and stabilizes C3B BB. Let us come to the third pathway, which is also a part of the innate system in the true sense, because its initiation or activation also is through the innate pathway, where no antigen antibody interaction needs to happen. This is the lectin pathway. Lectins are proteins that bind to sugars. Each one has different specificity. Now, MBL or mannose binding lectin has specificity for mannose, as the name suggests, and this protein is an acute phase protein. It is made or it's synthesized and secreted when there is an extensive inflammatory response or inflammation. MBL, short for mannose binding lectin, resembles the structure of C1q. It is not identical, but resembles. Now, MBL, once it is made or synthesized, binds to surfaces of cells and pathogen, and when it is bound in a fashion which is similar. Now, these heads bind to mannose present on bacterial surfaces, and you might remember again that bacterial surfaces have more mannose exposed on the cell surface than mammalian cells. The MBL is associated with serine proteases, and there are 4 of such subunits called two such MSP1 and 2, which form a tetrameric structure bound to the MBL. Now, MASP is the short form for MBL associated serine proteases. Now, you have a bacterial surface where the MBL is bound. This binding activates MASP1 and 2, the serine proteases 1 and 2, and this recruits C4 to the site, cleaves C4 to C4a and C4b, and the rest of the cascade is identical to what one sees in the classical pathway. A little bit with regard to the MBL or the mannose binding lectin, it has an oligomeric structure which could be 400 to 700 kilodalton. Subunits contain 3 identical peptide chains of 32 kilodalton to make that flowery structure, but it is known that MBL can form several oligomeric forms: dimers, trimers, tetramers, and so on. But it is the tetramer which is the minimum oligomer which can be activated for this, or rather can activate the complement cascade. Dimers and trimers have been seen not to be biologically active. What are the consequences of complement activation? one can imagine immediately that since the classical pathway is dependent on the antigen antibody interaction, and since there are enzymes involved, there would be an amplification of the humoral response. So, the classical complement components or cascade amplifies the humoral response, converting it to destruction of invading cells. Antigen and antibody binding can bring about inactivation of the protein by way of complex formation, which is or immune complex formation, which can be thrown out of the body by phagocytosis. However, antibodies do not kill themselves. So, here is the strategy by which antibodies can recruit the complement and trigger the cascade, so that membrane attack complexes are formed and they can kill broad spectrum of microbes, red blood cells and nucleated cells. We should not forget, and though we are not going to talk too much in detail with respect to the split products that are formed C3A, C4A, C5A, these are called anaphylatoxins. They bind to receptors on mast cells and basophils and induce degranulation of the preformed granules of these cells, which have histamines and mediators of inflammation. The split products also induce monocytes and neutrophils to extravasate. Complement components can also opsonize, that is, bacteria surfaces since they bind to C3b and quite stably, then the opsonized bacteria, which would mean bacteria that have bound to C3b, can now be recruited to phagocytes which express complement receptors, complement component receptors CR1, CR3, CR4, which are the receptors for each of the complement components. Now, bacteria bound to C3b are recruited to CR1, for example, to the phagocytes, and this enhances phagocytosis, thereby mediating clearing of immune complexes. In fact, this complement cascade, which brings about clearance of the immune complexes, is something that is an important function, because in diseases where this cannot take place would lead to hypersensitive reactions, and we will come to that in a later lecture. Clearance of the circulating immune complexes, how does that happen? You have the complex formation, antigen antibody interaction. This now brings about activation of the complex such, what is not depicted here is the formation of the C1q binding here, and then C4b 2a, and then formation of C3b. So, we come directly to C3b, which would be attached to this site, and the C3b is here in red. The C3b has receptors on red blood cells as well as phagocytes. The red blood cells CR1, because you can imagine that the red blood cells are large in number and in circulation, and therefore, the complex can load onto the CR1 receptor on the red blood cells, and this RBC with its cargo now donates this complex to the phagocytes, because the CR1 phagocytes have many more CR1s and have good affinity, and phagocytes can also phagocytose this complex, and thereby this is in fact cleared from the system. RBCs then go to the spleen for their final destruction. Is there any regulation of the complement system? There should be, right, because after all complement cascade involves enzymes, and once these enzymes are triggered, there is a possibility that the complement cascade can go, the entire cascade can bring about hypersensitive reactions. There has to be a regulation of the complement system, therefore, so that bystanders are not hurt or normal tissue is not hurt. there are both soluble as well as membrane bound proteins which regulate the complement system. C1 inhibitor. These are also, all these components are present in serum because they are soluble. C1 inhibitor that causes dissociation of the R2S2 from C1q, that means the complex is constantly broken. So, the cascade cannot. C4b binding protein which blocks the formation of C3, it binds to the surface of C4b which should recruit C3, which should bind to C3, sorry, and therefore, now the enzyme is not allowed to act. Factor H blocks formation of C3 convertase by binding to C3b. Factor 1, it cleaves C4b, C3b. S protein binds soluble, you remember the C5b67, which should insert into the cell membrane, but does not because of the presence of S protein. And then also anaphylatoxin inactivator, which blocks anaphylatoxin activity. Membrane bound, there are molecules CR1-MCB, decay accelerating factor, HRF and MIRL, all these block. Now, the last two block C9 polymerization, and CR1 catalyzes cleavage of C3b, C4b, and decay accelerating factor dissociates the C4b2A or the C3 convertase. There are genetic deficiencies which have been described for each of the complement components, and homozygous deficiencies of early components exhibit marked increase in immune complex diseases such as SLE and glomerular nephritis. These patients also suffer from recurrent infections. Now, to summarize the complement system, the complement activation involves an enzymatic cascade generating various complement proteins that play a role in the clearance of pathogens and antigens. There are three pathways: the classical, the alternate and the lectin pathway. All of them have different ways by which they get activated, but converge in a common terminal sequence that generates the membrane attack complex. The importance of the system is realized, though it is a part of the innate immune system. The importance is realized from clinical studies. Persons with deficiencies of any of the components suffer from recurrent infection, and it has been found those with C3 deficiency are most prone. Because of its non-specific nature, the complement system requires elaborate regulation so as to inhibit damage to normal tissues, and all the inhibitors that I talked about in the last two slides are there to limit to an extent, but not to abrogate the complement cascade. Thank you.